Hi, I am Dr. Neela Bakore, and in this segment we are going to start with the process of spermatogenesis and oogenesis, that is gamete formation. This is the first step in sexual reproduction. Up till now we have seen the structure of male reproductive and female reproductive systems. Now we are talking about the first step of the sexual reproduction that is seen in human beings or most of the mammals. So, chapter is same, reproduction in humans, and we are going to talk about gametogenesis. We have discussed about this, that gamete formation takes place in male reproductive structure and male reproductive structure, that is testis, and female reproductive structure, that is ovaries, the primary sex organs. So, gametogenesis is of two types. Sperm formation is spermatogenesis and egg formation is oogenesis. So, after these processes, we will also see how the sperms are formed and what is the structure of it, and same is in the case of oogenesis, how this process takes place and what is the structure of the egg. So, now let us talk few more things about spermatogenesis. Spermatogenesis takes place in the germinal epithelium of seminiferous tubules. So, site is seminiferous tubules. In case of some mammals like bats, otters, llamas, the male animal shows testicular cycle. Testicular cycle is a phase which takes place during reproductive period. So, when they have their reproductive period, the testes, they enlarge, become functional and descend into the scrotal sac. So, during this cycle what happens is the testes enlarge, testes become functional, and they descend into scrotal sac. During the reproductive period, once this period is over, they will again become smaller, non-functional, and will ascend into the abdominal cavity. That means all the time when the reproductive phase is not there, at that time the testes are intra-abdominal, non-functional and reduced in size. Only during reproductive period there is a change which takes place. This is known as testicular cycle, during which the testes enlarge, they become functional and descend into scrotal sac. Spermatogenesis will take place, sperm formation, they will reproduce, and again in non-breeding season the reverse would take place. So, in these animals, that is bats, otters, llamas, the testes remain in the abdominal region during non-reproductive phase. During breeding season they show testicular cycle, and testes they descend into scrotal sacs. So, this is one category of animals. The second category of animals, it includes mammals which have testes in the scrotal sac always. Once, like we talked about in human beings, the testes descend into scrotal sac in the 7th month of gestation period. After that, they remain in the scrotum throughout the life. So mammals which have extra abdominal testis all the time. So there is no such period that this is breeding period when there would be testicular cycle, and non-breeding period where the testis would ascend into the scrotal, into the abdominal cavity. They permanently remain in scrotum. The animals which come in this category, the mammals rather which come into this category are human beings. So man, bulls, dogs, lion, tiger. These are the animals where the testes are always extra abdominal. There is a third category, those mammals in which the testes are permanently intra-abdominal. So mammals with intra-abdominal. The examples are elephants, whales, these animals, they have the testes in the abdominal cavity all the time. That means spermatogenesis takes place in the abdominal cavity only. In case of these three categories, the difference is in the first category where animals show testicular cycle, during non-breeding period the testes remain in the abdominal cavity. During breeding period they descend into abdominal cavity, descend into scrotal sac and become functional. Second category, all the time extra abdominal. Third category, all the time intra abdominal. So these are three categories of mammals. Spermatogenesis is a continuous process. That means it starts and then it goes on and on and on without any gap or pause. This we will be able to understand in a better manner after we discuss both spermatogenesis and oogenesis. Oogenesis is a discontinuous process. So once we understand what exactly is this discontinuous process, we will be able to understand this particular thing in a better manner. So we will come back to this point when we discuss oogenesis. In human beings, spermatogenesis is completed in 74 days. In humans. In human males, once it starts at puberty, and once that complete process begins from the germinal epithelium, sperm formation to sperm formation is completed in one go, and that is completed in 74 days. It is a continuous process. Now some basic things about spermatogenesis. Now let us take the process. It takes place in the seminiferous tubule and the site is germinal epithelium. So if we are talking about these cells, the cuboidal cells, and here there are those pyramid-like cells which are called the Sertoli cells. All other cells, the cuboidal cells, they will undergo this process. So if we have to divide this process of spermatogenesis, site we have seen, the process can be divided into two steps. Number one, formation of spermatids, spermatid formation, and step number two is spermiogenesis. These are the two steps of spermatogenesis. It is completed in two parts. The first part is spermatid formation and second part is spermiogenesis. Spermatid formation is completed in 3 steps or 3 phases. First is known as multiplication phase. Which is simple mitotic divisions. This is followed by growth phase. As the name tells about what is going to happen in this process is simply growth, and third is maturation phase. During maturation, meiosis would take place. So let us write down what is going to happen in multiplication phase, mitotic divisions. In growth phase, growth, and in case of maturation phase, meiosis. So now let us see how all these changes take place in the germinal epithelium. Germinal epithelium of seminiferous tubules. The cells of this germinal epithelium, they will undergo the first phase of the first part. For our reference, let me write down. Spermatogenesis, 2 parts, that is spermatid formation and spermiogenesis. And the 3 parts or the 3 phases of spermatid formation: first, multiplication, then growth, and third, maturation. So we are talking about the first phase of the first part, that is multiplication. These cells, they start to multiply. Now here we have to visualize it a little bit. Everything is happening at the same place. That means this cell will multiply, 2 daughter cells will be formed because it is mitotic division. So all of them will be clustered in this region. But when we have to show the stages, we will draw one stage here, the next stage, third, fourth, and so on. So that does not mean that the cells are moving away from the epithelia, because they have to get nourishment from these Sertoli cells. So they will remain attached to the Sertoli cells. So now these germinal epithelial cells, suppose now I am showing the cell separately, but it is still here attached to the membrane. It undergoes mitotic division. This is mitosis. The cells are diploid. The germinal epithelial cells are diploid. These cells, they are called sperm mother cells or spermatogonia. So when it divides, it will again produce diploid cells because the division is mitotic division. These cells again will divide because this is a continuous process. They will go on multiplying. So again mitosis, and there would be many spermatogonia. So these cells, they are called spermatogonia, which means sperm mother cells. These are the cells which are going to give rise to the sperms. These cells, they are of two categories. They are called type A and type B. Type A act as stem cells and they undergo multiplication. So when they multiply, they will produce again half A and half B type of cells. So they will divide to produce more spermatogonia. Divide to form more spermatogonia. The type B cells, they will enter the next phase. So they participate in the process of spermatogenesis, that is growth phase. So now they will enter the next phase. So half or some cells will act as type A cells, half or some cells will act as type B. Type A will go on dividing, like here we saw this just underwent division. So type A will go on doing the same thing. Type B will enter the next phase, that is the growth phase. So what is happening in this part? If we have to just write down the phase, this is multiplication phase. This entire phase is multiplication. Now these spermatogonia undergo the next phase, that is growth phase. Growth is accumulation of proteins, RNAs and all these things. In case of egg formation, the stages are going to remain same, but what happens in growth of an egg, normally the size of the egg increases because of yolk accumulation. In case of sperms, there is no yolk. So the size is like just doubled or maybe 4 times, so not much because there is no yolk formation. Only accumulation of proteins and RNA and all those things is required. So, or rather takes place. So here it increases in size but not that much, like double. So this part is growth phase, where the spermatogonium has grown by accumulation of proteins and RNA. This cell is known as the grown cell now is known as primary spermatocyte. Spermatogonia are diploid. Here only growth has taken place, no division. So this is also diploid. Now this primary spermatocyte undergoes the next phase, that is maturation phase. And maturation phase, we said, is meiotic division. We have done cell, we have understood cell in class 11. In meiosis, we know that meiosis is completed in meiosis 1 and meiosis 2. So this cell will undergo meiosis 1. Meiosis 1 is reductional division. So the cells which are formed here as a result of meiosis 1 which is a reductional division. These cells will be haploid cells and they are known as secondary spermatocytes. Why are they haploid? Because the primary spermatocyte undergoes meiosis 1, which is a reductional division, and from one primary spermatocyte we now get two secondary spermatocytes. Both these spermatocytes would undergo meiosis 2. Meiosis 2 is equational division, it is similar to mitotic division. So here there would be 4 cells obtained, 2 from each secondary spermatocyte, and the chromosome number will remain same as that of the secondary. So here this is haploid, haploid, haploid and haploid. So these cells, they are called spermatids. This is the first part, spermatid formation, which is completed in multiplication phase, then growth phase and maturation phase. So now this, what has happened here from the primary spermatocyte to spermatid formation is maturation phase. Maturation phase is meiosis. So from one spermatogonium, that is one sperm mother cell, 4 spermatids are obtained. These circular haploid cells which are formed, they do not resemble the sperms. The sperm structure that we have seen or heard so far is some structure with a little head-like part and a long tail. So this structure, that is spermatid, has to change into the sperms, and now it will change into this sperm. So these structures which are formed, they would be called sperms, and the process which is required for conversion or changing a circular spermatid, circular haploid spermatid into a functional sperm is known as spermiogenesis. So this process which is taking place, that means we are talking of this change, is known as spermiogenesis. Spermiogenesis includes the changes which take place in a spermatid for sperm formation. So now from one spermatogonium, we will see the details of what all happens during spermiogenesis, but from one spermatogonium, 4 sperms will be produced, because the division is first mitotic, then growth, then meiosis, which results into formation of 4 haploid spermatids which get transformed into the sperms by the process of spermiogenesis. Now let us see what all changes take place between the stage that is called spermatid and the sperm. This process is simple, and we will talk about the types of sperms. There are 2 types of sperms produced, but once we understand these changes, then we will take that up.